Hey guys, welcome back to One Stop Biology. So today we are going to start off with a new chapter of class 12th, which is molecular basis of inheritance. Now in this chapter we are going to basically cover all the different kind of genetic materials that we basically have in animals or plants, right? So we are going to discuss about DNA, RNA, how it is transcribed, how it is translated into proteins and all that, right? So we'll start off with the DNA. So guys, basically in the last chapter of genetics, the first chapter, we studied about how Mendel basically, experimented on pea plant, and then later on various scientists, they experimented on certain plants and animals, and they got one thing, that there is a factor which is inherited from one generation to another, and we call it that factor now as gene, right? And in one of the chapters of class 11th, we have also studied that nucleic acids are polymers of nucleotides, and these nucleic acids are basically the basis of formation of DNA or RNA, right? So basically, what exactly is DNA and RNA? Now, DNA, which is also known as deoxyribonucleic acid, and RNA, which is ribonucleic acid. Both are different type of nucleic acids, as the name suggests. Both the nucleic acids are found in the living system, right? So basically, DNA is something which is considered as the genetic material in most of the organisms. DNA acts as the genetic material in most of the organisms, and in some organisms like some viruses, RNA acts as the genetic mes- material, right? And it functions as a messenger, right? So what we are going to do is we are going to first start off with DNA, its structure, the replication, and then we'll see the process of making of or RNA from DNA. We'll see what is the genetic code all about, right? And how the sequence of amino acids basically determines the proteins, and how the RNA is translated into a protein as well. So what exactly a DNA is? DNA is a long polymer. It is a long polymer of what? Of deoxyribonucleotides. As mentioned here as well, DNA is the long polymer of deoxyribonucleotides, right? Now, the length of this DNA is usually defined as the number of nucleotides which are present in it, right? So the number of nucleotides or base pairs which are present in it. Say for an example, a bacteriophage. A bacteriophage has 5386 nucleotides. In the same way, a bacteriophage lambda has 48,502 base pairs. E. coli has 4.6 into 10 to the power 6 base pairs. And haploid content, remember haploid content is mentioned here, although humans are diploid. So half of the content of human DNA has 3.3 into 10 to the power 9 base pairs of nucleotides, right? So this depends on basically the length of DNA depends on the number of base pairs or number of nucleotides which are present in them. And DNA is nothing but long polymer of deoxyribonucleotides. Now, what is the structure of this nucleotide chain, of this polynucleotide chain? So basically, the structure of this polynucleotide chain says that the nucleotide has three components, right? So any nucleotide has three components. So we have a nucleotide, It has three components. One is a nitrogenous base. So nitrogen base is there. Then we have a pentose sugar. We have a pentose sugar. And then we have a phosphate group. So there are three parts to a nucleotide, right? We have a nitrogen base, we have a nitrogenous base, we have a pentose sugar, and we have a phosphate group. Now, this pentose sugar in case of RNA is ribose sugar. In case of RNA, it is ribose sugar. In case of DNA, it is deoxyribose sugar. Right? Now, again, let's first talk about the nitrogenous bases. So, the nitrogenous bases are of two types. They are of two types. One is purine, and the other is pyrimidines, right? So we have two types of nitrogenous bases. One is purine and the other one is pyrimidine. Now again, purine bases are of two types. Purine bases are basically two. One is adenine, the other is guanine. In the same way, pyrimidine base again are two types, or say three types. We have cytosine, we have uracil, and we have thymine. So we have cytosine, uracil, and thymine. Guys, you'll have to remember all of this, right? So now basically what happens is cytosine here is common in both DNA and RNA, right? Thymine is present in DNA and uracil is basically a replacement of thymine in case of RNA, right? This is present in DNA, this basically RNA, and this both DNA as well as RNA. So we have always four types in DNA and four types in RNA. In DNA, it will be a combination of adenine, guanine, cytosine, and thymine. In case of RNA, it will be adenine, guanine, cytosine, and uracil. So thymine is basically replaced by uracil in case of RNA, right? So this is what we study about the nitrogenous base. Now, how it is linked, let's study that as well. That how it is linked to the bases or the phosphate group, because it is a chain, right? So they'll be joined to one another, right? So a nitrogenous base is linked to OH. It will be linked to the OH group of first carbon of pentose sugar, right? So it will be linked to OH group of first carbon of pentose sugar through a N-glycosidic linkage, right? So nitrogenous base is linked to OH group of first carbon of pentose sugar through N-glycosidic linkage, and this linkage along with the sugar forms nucleoside such as adenosine or deoxyadenosine. In case of guanine, it will be guanosine or deoxyguanosine. In the case of cytosine, it will be cytidine or deoxycytidine. In the case of uracil, it will be uridine or deoxyuridine. In the case of thymine, it will be thymidine or deoxythymidine, right? So when a phosphate group is linked to OH of fifth carbon of nucleoside, now this is basically combining the nucleotide with the pentose sugar. Now this forms the nucleoside. Now the entire nucleoside will be linked to the phosphate group. The phosphate group is linked to again OH group of fifth carbon of nucleoside. It is attached to fifth carbon of nucleoside through what? Through phosphoester linkage. Through phosphoester linkage. Then a corresponding nucleotide or deoxynucleotide is formed. Then a corresponding nucleotide or deoxynucleotide is formed. Now, two nucleotides. So basically, we have studied about two types of linkages. The base, the nitrogenous base is linked to pentose sugar through N-glycosidic linkage. Then the nucleoside is linked to phosphate group through phosphoester linkage, right? Now, two nucleotides. These nucleotides are formed, right? Now, two nucleotides are linked to through 3 and 5 phosphodiester linkage. Phosphodiester linkage to form a dinucleotide. And then more than two nucleotides, many nucleotides are joined to form a polynucleotide chain, right? So I hope you understood how a polynucleotide chain is formed, right? Now a polymer formed has one end, as in like one end of that polymer has a phosphate moiety, right? Which is at 5 end of sugar. So you have one end has phosphate at fifth carbon, which is known as 5' end of polynucleotide chain. Now the other end of that polymer has sugar, right? Now this sugar has free OH and this is present at third carbon. This is present at third carbon and that is why it is known as 3' end of polynucleotide chain, right? So again remember that the polynucleotide chain, so this is the polynucleotide chain. This is basically one nucleotide. This is the other nucleotide. This is the third nucleotide. This is the fourth nucleotide. So you can see there is a phosphate group, there is a pentose sugar, and then you have the nitrogenous base, right? This keeps changing. So this is a polynucleotide chain. One end has phosphate group at fifth carbon. The other end has free OH at third carbon. So the first end is 5 dash, second end is 3 dash. Now, backbone. Remember that backbone of a polynucleotide chain is formed due to sugar and phosphate. So, the backbone of the polynucleotide chain is nothing but the pentose sugar and the phosphate, right? Now, the nitrogenous bases linked to sugar moiety project from the backbone. So, the nitrogenous bases are basically projecting from the backbone. They are attached to the pentose sugar and they are projecting from the backbone, right? Now, in the case of in RNA, what happens is every nucleotide residue has an additional OH group present at second position in the ribose sugar, right? So, what happens is there is an additional OH group present at second position of the ribose sugar, and then in place of thymine, you have uracil in RNA, right? Now, let's talk more about the DNA first. So, basically, DNA as an acidic substance was present in nucleus and it was for the first time identified by Friedrich Mischer. And this was identified in the year 1869. And he named it as nuclein. And then later in 1953, James Watson, guys, these names are very important because they gave the 3D model of DNA, Watson and Crick model. So James Watson and Francis Crick, based on X-ray diffraction data, which was produced by Morris Wilkins and Rosalind Franklin, they proposed double helix model of DNA. They proposed what? They proposed double helix model for structure of DNA. So basically they gave Watson and Crick model of DNA, which was a double helix model, right? Now, one of the, hallmarks of their proposition was base pairing between two strands of polynucleotide chain. So there are basically DNA is double helical. There are two strands running parallel and there is base pairing. There is pairing of the nucleotide base among them. There is the pairing of nitrogenous base among the each other. We have four bases, so they are basically paired in a sequence, right? So this proposition was based on basically observation of Erwin Chargaff that for a double-stranded DNA, the ratio between adenine and thymine, so adenine is denoted as A and T, thymine, and guanine and cytosine are constant and equals 1, right? So what he said is Erwin Chargaff basically said that the ratio between A and T and G and C is always constant and that equals 1, right? Now the base pairing confers that polynucleotide chain are said to be complementary to each other. The polynucleotide chain, the two polynucleotide chain which forms the DNA are complementary to each other. And that is why the sequence of bases in one strand is known, then the sequence in the other strand can be predicted because it is complementary, right? Now, if each strand from a DNA acts as a template for synthesis of a new strand, say we consider one of the strand of DNA as template for synthesis of a new strand, the two double-stranded DNA later on produced would be identical to the parental DNA molecule. And because of this, the genetic implication of structure of DNA became very clear because both these strands or the new strands formed will also be complementary and they'll be identical to the parent DNA. Now, the salient features of double helix structure of DNA was that one, it is made up of two. First one is that DNA is made up of two polynucleotide chains, right? Where what happens? The backbone consists of sugar and phosphate, right? And the bases project from them, from the backbone. The bases project inside. Now, the second feature is that two chains have anti-parallel polarity. They are anti-parallel. So, say if one chain, say this is the first chain, if this runs 5 to 3 end, right? The other chain will be anti-parallel. So the other chain will be running towards the opposite end, right? So this is the second feature. Now the third feature is that bases in two strands, the bases which are found in two strands, they are paired through hydrogen bonds. They have hydrogen bonds forming base pairs, right? So always now remember. So another important point that we got is that the base pairs are joined by hydrogen bonds. And the pair is between adenine and thymine It is always between adenine and thymine and guanine and cytosine in case of DNA, guys, right? Now, what happens is adenine and thymine has two hydrogen bond. They have two hydrogen bond. They have two hydrogen bond. Guanine and cytosine have three hydrogen bonds. They have three hydrogen bond, right? And this is from the opposite strand. So basically, if you see here, this is one strand and this is the other strand. If you see the complementarity, it is opposite. This is running 5 to 3, this is running 3 to 5, right? Now here are the nitrogenous bases. So see how they are attached to the opposite strand. So there are two hydrogen bonds between adenosine and thymine, and there are three hydrogen bonds in between guanine and cytosine, right? So this generates what? This basically generates approximately uniform distance between the two strands of the helix. Remember that this hydrogen bond attachment between the nitrogenous bases among the opposite pairs, opposite strands, maintains uniform distance between the two strands of the helix. So this is the third feature. Now the fourth feature is that the two chains, the two strands are coiled in a right-handed fashion. The two strands, I'll repeat it again, are coiled in a right-handed fashion, and the length of each helix, the pitch of each helix is 3.4 nanometer, right? 3.4 nanometer. And there are 10 base pairs. There are around 10 base pairs in each helix, in each turn. So the distance between one base pair in a helix is approximately 0.34 nanometer. It is approximately 0.34 nanometer. So the DNA helix is right-handed, right? Then the length of one helix, one turn is 3.4 nanometer, and there are around 10 base pairs. So the distance between two base pair is around 0.34 nanometer. This is the fourth feature. Now the next one is that the plane of one base pair, the plane of one base pair stacks over the other, right? In a double helix. This, in addition to hydrogen bonds, confers stability of the helical structure. So basically, what it means is that one base pair stacks over the other, right? And basically, this stacking and then the hydrogen bonds which are present between the base pairs gives the helical structure stability. So if you see this, a DNA double helix, these are the base pairs, right? So if you can see here, basically you can see that they are stacked, right? So they are stacked, and this ribbon kind of thing that you see in the structure is nothing but the backbone, the sugar phosphate backbone, right? So this is the exact structure, right? Now let's see what Francis and Crick said. Either they gave some more clarification, right? So Francis and Crick also proposed that the central dogma is molecular biology, which states that genetic information flows in the direction as shown here. It flows starts from DNA, which keeps on replicating itself. DNA transcribes into mRNA, messenger RNA, which it translates into proteins. So this is the central dogma. What is the central dogma? In molecular biology, it states that genetic information flows from DNA to RNA and then from RNA to protein. Now, from DNA to RNA, it is known as transcription. From RNA to protein, it is known as translation, right? Now, in some viruses, this flow of direction is reverse, which is from RNA to DNA. In some viruses, right? Now, let's see the packaging of DNA helix as well. Now, the length of the double helix, remember the length of a double helix of a typical mammalian is around 2.2 meter. It is around 2.2 meter. Now, in prokaryotes such as E. coli, they do not have a defined nucleus. DNA is not scattered throughout the cell. Even in prokaryotes, though they do not have a defined nucleus, but DNA is not scattered, right? So basically, DNA are negatively charged. Now, DNA being negatively charged is held with some proteins. These proteins are positively charged, right? And basically, this region where the DNA are held with proteins, this is known as nucleoid, right? Now, DNA in nucleoid is organized in large loops which are held by proteins, right? Now, what happens in the case of eukaryotes? So, in the case of eukaryotes, there is a set of positively charged basic proteins. So since DNA are acidic in nature, you need proteins which can bind them so that the protein has to be positively charged. So in eukaryotes, there are proteins which are basic in nature and positively charged. These are known as histones, histone protein. These are histone protein, right? Now, a protein acquires charge depending upon the abundance of amino acid residue with charged side chain. The histone protein acquires charge depending upon the abundance of amino acid residues, right? Now, in case of histones are rich in basic amino acid, right? And these amino acids are lysine and arginine. They both carry positive charge in their side chain. So histone proteins are rich in basic amino acids like lysine and arginine. Now these histones are organized to provide a unit of 8 molecules, which is known as histone octamer. So basically say this is one histone protein. Now this histone protein are organized along with 8 molecules of histone, and this forms a histone octamer, right? Now the negatively charged DNA is wrapped around the positively charged histone octamer to form a structure which is known as nucleosome, which is known as nucleosome, right? So basically what is happening is that the single histone protein combines with 7 more and it forms histone octamer, and then this histone octamer, the negatively charged DNA is wrapped around, right? And this complete structure where negatively charged DNA is wrapped around histone octamer is known as nucleosome. It is known as nucleosome. So if you see the structure here as well of the nucleosome, in this structure what you'll see is that this is the histone octamer. This circle that you see, sorry, yes, this circle that you see is the histone octamer, right? And then the DNA is coiled around the histone octamer, and this entire structure is known as nucleosome. That is known as nucleosome. Now, a typical nucleosome usually contains 200 base pair of DNA helix, right? So, on one nucleosome, you have 200 base pairs of DNA helix, right? Now, these nucleosomes constitute the repeating unit of a structure, which is known as chromatin. This chromatin is thread-like stained bodies which are seen in the nucleus. So basically, these nucleosomes keep on repeating, right? And that repeating unit is seen is in nucleus as chromatin, which is a fiber, which is so we call it as chromatin fiber, right? Chromatin fiber. These are thread-like stained bodies, right? Now, nucleosomes in chromatin are seen as beads on string. So you have thread-like structure and you can see beads on string, which are nothing but nucleosomes when you see it under a microscope, right? Now, these beads on string structure, remember that these beads on string structure in chromatin is packaged to form chromatin fibers, which are further coiled and condensed. So what is happening is basically, if I draw it in a go, you have these octamers, you have DNA being coiled, right? So this is somewhat like the beads on string. And then this is again coiled. So now this entire structure is again coiled and condensed. And this happens at metaphase stage of cell division. And this forms a chromosome. So basically, if you see this structure of a chromosome, this is nothing but condensed coiling of DNA or the nucleosome, DNA with the histone protein. This is basically condensed and compressed in a in the form of chromosome. So these nucleosome forms chromatin, which forms chromatin fibers, and those are further coiled and condensed to form chromosomes. Now, packaging of chromatin at higher level requires additional set of proteins which are known as non-histone chromosomal proteins, NHCs, right? So, further coiling needs additional set of proteins which are non-histone chromosomal proteins. Now, in nucleus, there are some regions of chromatin which are loosely packed and stains light. These are known as euchromatin, right? So if they are loosely packed and they stain lightly, they are known as euchromatin. If they are densely packed and they stain dark, they are known as heterochromatin. Now, what is the function of euchromatin and heterochromatin? Euchromatin transcription basically is transcriptionally active chromatin. The light and loosely packed one is transcriptionally active chromatin, right? And heterochromatins are transcriptionally inactive chromatin. They are transcriptionally inactive chromatin, right? So, guys, I'll repeat one thing again as we have just finished all about DNA. So, basically, we have DNA in the form of helix. Then we add histone octamer and we form nucleosome. Now these nucleosomes form chromatin. These form chromatin fibers. Now these are even more condensed to form chromosomes, right? So this is how the DNA is formed, how the DNA was discovered, what is the structure look like, and what are each and every components of DNA, guys. This is a very, very important topic. I'll repeat it again. It is very, very important topic from your boards and NEET point of view. And I would request you that after watching this video, please go through the NCERT topic of DNA at least 3 times, and please do let me know if you have any doubts. Please mention your doubts in the comments below. You can also ask your doubts in the WhatsApp number that which is given in the description of the video, and I'll try and clear your doubts immediately. And if you did like the video, please do not forget to comment that yes, you have understood the video and you like the video. And please do not forget to like the video and share it with your friends. And yes, if you are new to the channel, please do not forget to subscribe to the channel. Thanks, guys. Bye bye.